Hi, good morning everyone. I'm Annie from National University of Singapore, and I'll be presenting on the spatial temporal correlates of age-related gene expression and cortical morphology. I'll also be having a poster session today at 1:45, and if you have any questions, feel free to look for me at my poster. I'll be happy to answer them. The concept of developmental origins of aging has received great attention. It suggests that brain aging could be the reverse process of brain development. In other words, events that's critical in early life could have an impact on later life with regards to neuronal and synaptic loss. Independent evidence from neuroimaging and genetic studies suggests a possible link between development and aging, which could potentially lend support to this concept. From the neuroimaging study, it has shown that brain volume loss mirrors brain volume gain in development. And from transcriptional study, gene expression changes in early life are reversed in aging in the prefrontal cortex, and in recent study is shown in temporal and parietal cortices as well. Such a reversal trend of gene expressions across the lifespan is suggested to be a genomic reflection of responses to cellular changes such as oxidative damage, or perhaps possible consequences of these changes. Now, genetic factors are a dominant force in explaining variants of many measures of brain structure throughout the lifespan. However, it remains unclear how gene expression profile relate to changes in cortical volume. And are there any biological process or health-related traits that can be targeted in early life to prevent aging in later life? Or are there any biological process or traits that can be targeted to prevent aging only in later life? Our aim here is to integrate the gene transcriptome and brain morphology data across lifespan, and to investigate the spatial temporal correlates of the gene transcriptome and brain morphology in discovering the biological process and traits across the lifespan and in aging. Data curated by Kang in 2011 were used because it contained more time points and more brain regions as compared to other transcriptomic data sets. There were total of 26 donors aged 3 to 80 years old, and 17 over 1,000 genes across 11 cortex. In terms of the brain morphology, we utilized over 2,000 over T1 weighted MRI scan from the healthy subjects. And this data was processed in free server. Cortical volume that corresponds to the region that was being sampled in the Kang data set were then extracted as shown in the figure. We first look at the age effect on the cortical volume in relation with the age via regression model, and it shows that all the 11 cortical volumes show these up-down patterns with age. Likewise, we also show the standardized volume, where the standardized cortical volume will average within each time point and also between left and right hemisphere. They will then plot it again on the same figure in order to display this up-down pattern for all the 11 cortical regions. In my right, age effect on the gene expressions value of each gene in each brain region were also analyzed using the quadratic regressions, and we have identified genes that show consistent temporal patterns across the 11 cortical regions. Here we use one of the genes, MAPK1, to display the significant up-down expression patterns across all the 11 cortical regions. Similarly, we use another genes, which is the HYAL2, to represent the down expression patterns across the 11 cortical regions. In total, we identified around 198 genes out of the 17 over 1,000 genes that show up-down or down-up expression patterns with relations to the age. Subsequently, we did a Pearson correlations between the gene expressions of the up-down and down-up genes with the cortical volume across lifespans, and these were repeated for all the 198 genes. Through this analysis, we have identified 155 genes that are spatially temporally correlated with the cortical volume. Using the 155 genes, we did a protein-protein interaction network enrichment analysis to show that this group of genes were biologically connected. And in addition, we also tried to identify the hub genes based on the degree calculations, whereby for every gene, we calculate the number of connections it has with the rest of the genes in the PPI network. Genes that were highlighted in red are considered as hub genes because they have more degree and at least one standard deviation above the mean degree of the whole thing. And all these red hubs are considered the biomarkers of the brain development and aging. We then move on to look at the cell type, biological process, as well as the brain disorder that these genes might play a role in. We first ask what cell types are these genes expressed, and we observe that majority of the genes are expressed in neurons, as shown in the dark brown. We then ask in which biological process are these genes involved, and we observe that they were involved in the synaptic transmissions and plasticity, as indicated in purple. Here in the figure, each green actually represents a process that's related to a synaptic function, such as regulations of the synaptic plasticity, neurotransmitter receptor, and transmissions, just to name a few. They also involve in signal transduction, such as the glutamate receptor signaling pathway, as well as the calcium-mediated signaling pathway. They also involve in metabolic process, such as the amyloid beta metabolic process. And they were enriched for neurodevelopmental and neurodegenerative disorder. The hub genes identified as earlier on also showed to be predominantly involved in the synaptic transmission and plasticity, although there are also a few genes that are involved in other processes as well. Linkage-based equivalence score regression were used to determine whether genes involved in the brain morphology across lifespan were associated with traits related to neuropsychiatric conditions, neurological, development, and health-related traits. And we found that the genes that involved in the cortical volume across lifespan were highly enriched in the bipolar disorder as well as the neuroticisms. So far, across the lifespan, we have identified biological process that's related to synaptic plasticity and transmissions, as well as signal transduction. And traits involved were related to the mood traits, such as bipolar disorder and neuroticisms. They were also highly expressed in neurons, and they were enriched in neurodegenerative and neurodevelopmental disorder. Apart from examining these across lifespan, what we want to do is to understand whether there are any unique biological process that is happening in aging only, and therefore we perform the spatial temporal correlates using subjects from 55 years old and above. Using the aging data set, we observed that the same biological process that was identified using the lifespan data set includes synaptic transmission and plasticity, as well as signal transduction as shown in the blue. However, we also observe biological process that's uniquely associated with the aging data set. And this includes predominantly the metabolisms, autophagy process, cell development, and migration. In addition, enrichment for traits were again performed using the aging sample, and they were finally to be enriched in the bipolar disorder. Here's a summary for the result as shown using the aging data set only. And in comparisons with the results from lifespan data set, there was similar process being observed, such as the synaptic plasticity and transmission, as well as the signal transductions. The result for the aging dataset additionally show process that's related to cell development and migrations, autophagy and metabolic process. In terms of traits, genes that were involved across lifespan and in aging only were found to be enriched for traits that's related to mood. And in the cell types, genes that were involved across lifespan were highly expressed in neurons, while the genes that were involved in aging were expressed in both neurons and astrocytes. And lastly, in terms of the brain disorder, those genes that were involved across lifespan were mostly enriched in neurodegenerative and neurodevelopmental, while those genes that's involved in aging only were highly enriched in neurodegenerative disorder. In conclusion, neurons play a large role across lifespan, while astrocytes play a large role in the aging brain. The simulations of the cell development may help prevent brain aging, and enhancement of the synaptic transmissions is a potential common target in development and aging that could be used to prevent brain aging later in life. And in terms of our trait analysis, it might show that being happier in life could help you to age better, something that we can take care of and do in our life ourselves. And lastly, I'd like to thank all my collaborators as well as members from the Laboratory for Medical Image Data Sciences at the National University of Singapore.